Right. All right, just pipe down, we can get started. I got used to the silence that we get now. All right, so let's start out by, I've got everybody taking the test that I believe is going to take the test. So I have four questions here that we'll go through. These were the most missed questions on the exam. All four of these were somewhere between 30 and 40% of you still got right. Let's see, I got one more person coming in here. There we go. So there wasn't any one question that everybody missed. Actually, there was one question that everybody missed, and it was because I made a mistake on the answer key. So I fixed that and adjusted everybody. Again, plus you all had a 1% curve, so not a lot this time, but the mean was pretty close to a 75, so that was a pretty reasonable mean. All right, so these organisms are saprophytic. That's just straight terminology. Saprophytic means that the organism absorbs nutrients from dead organisms. So that answer would be fungi. This question, I believe, was the most missed one. This was a real curveball. I expected this one to be hard. I didn't have anybody come up and ask me and say, but none of these are the correct answer. The first time I read this question, that's what I thought. I didn't make this one up, but I like it because it's one that really makes you think. All right, so you know the three domains are the archaea, the bacteria, and the eukarya. Which of all these structures, which of them are present in all three of those domains? The correct answer here is the 70S ribosomes and plasma membrane. You know that they all have a plasma membrane. The 70S ribosomes, of course, in the eukaryotes are going to be present in the mitochondria and the chloroplast. So that one was kind of mean, but none of the other ones would have made sense. So suppose you're trying to visualize the movement of bacteria flagella in a live bacteria in live bacteria cells. What type of microscopy is going to be best? Well, if you want to, if you're trying to see live bacteria cells, that means you have to use some kind of microscopy where you're not staining them. All right, so confocal uses ultraviolet lasers and dyes. Of course, any kind of electron microscopy is going to kill the specimen. Fluorescence, again, dyes that fluoresce. That narrows it down to bright field and phase contrast. We talked about how you can see live cells in bright field, but the contrast is poor. So it's very difficult. And that's the whole reason that we have this special kind of microscopy, so we don't need to stain cells and we can still get good contrast there. You could also use dark field, but I didn't give that as one of your choices. So, all right, and which of the following is true concerning the causative agent of malaria? When I said causative agent, I think that threw a few of you. I just meant I didn't want to say protozoa because in the first answer here, I said the fungus. Okay, and Plasmodium is not a fungus, so that's why that one is not true. The correct answer here was growth and maturation of parasites in red blood cells is when we see the illness in humans, the sweating and the cold stage and all that. Okay, we know that the infection doesn't kill mosquitoes, if only it did. And the trophozoite stage is actually in the human red blood cells. That's the motile feeding stage. Okay, not in the mosquito. All right, so I would encourage you to come and talk to me. Few of you have come to see your exam, not too many. I will be in my office all morning tomorrow. If my office hours don't work for you, you might be able to find another time. I'm not available after class today. I have another appointment, but okay, so it's always good to come see what you missed. All right, so thinking back what we accomplished last lecture, we finished up our metabolism, talked about the difference between anaerobic respiration and fermentation. Those are two processes that are easy to get to kind of lump together because neither one of them require oxygen. They're quite different processes. One's a respiratory process, and of course, fermentation is not. Okay. And then we started our segment here on I almost said geometry, genetics, and we talked about the structure of DNA. Hopefully that was review. Talked about how DNA is semi-conservatively replicated, the enzymes that are necessary for it to be copied, to make a copy of itself. And also a little bit about the flow of information. So if DNA is copied, we call that replication. When we make RNA from DNA, we call that transcription. And then of course, it's the messenger RNA transcript that gets read at the ribosomes, and we call that translation. All right, so we're going to talk a little bit more detail about transcription than what you'll find in your book. I just don't find some of the figures in there to be terribly satisfying. So I'm going to show you a few things that are not in the book. But the three events of transcription, we're going to spend some time on initiation. That's when the main enzyme that's polymerizing the RNA, RNA polymerase is going to bind to the DNA. So that's a whole process in itself. Elongation is just that RNA polymerase moving down the DNA transcript and making more RNA. And then termination, there's a couple different ways that can be accomplished. So we saw in replication, pretty much the whole DNA is copied. So we didn't really talk about a starting place. Well, the starting place we called the origin, but there wasn't a whole process for starting and stopping just because the whole thing was copied. But in transcription now, we have only certain parts of the DNA being made into RNA. So there's, again, a whole process about where that starts and where that stops. All right. So a little terminology that I find always to be confusing. This is a figure that you won't see in your book, but it's talking about here's our RNA polymerase down here that's moving along the DNA here. This is just a blow up in the box at the bottom of, kind of a representation of the DNA here up at the top. Okay, so the RNA polymerase is moving down the DNA. And some terminology here. This strand of DNA that's being copied, right? Only one strand of the DNA is being copied, not the other. The strand that's being copied, we refer to that by several different names. We can call it the antisense strand or the negative sense strand. And that makes sense if you know that the RNA, the messenger RNA that's read at the ribosome, we always refer to that as plus sense RNA. And that's kind of important to think about because as we get into talking about viruses next week and how viruses take over our cellular machinery, the virus has to get their nucleic acid in the form of plus sense RNA because that's the only kind that our ribosomes will recognize. All right, so there are viruses that start out with negative sense RNA and they have to have a way to get that into positive sense RNA. All right, so this RNA that's being transcribed, we would call it plus sense RNA or so messenger RNA that gets read at the ribosomes is always plus sense. So if you know that, then it makes sense that this strand that's being copied would be the negative, because when you're copying nucleic acid, the plus sense would be the complementary bases or whatever that goes with the negative sense. Okay, when we copied DNA, we didn't get the same bases as what was on the template. We got the complementary sense, right? So the other name for this antisense strand, we could also call it the template strand, right? So if you're using something as a template, it means that's your guide. All right, if you're making up a resume, you might use a resume template to kind of help you get started. All right, so that kind of makes sense, at least to me. That's our template strand. What about this strand, the other strand of DNA? That could be the template strand if that was what was being copied. But in this case, it's the lower strand that's being made into RNA. And so we would call this upper strand of DNA the plus sense, the plus or the sense strand, because obviously the other strand is the minus, so this opposite would be the plus. And the other term that we use for that, the one that's not being copied, is the coding strand. And that might, to me, that doesn't make as much sense until I kind of write in some base pairs here. So let's say our messenger RNA, GGATU. All right, so I can't write small enough to make their little dash marks there. Say that's the messenger RNA. So the template strand here, the complementary bases would be CC. Whoops, I can't have a T and a U. Let me erase that. Nobody said, hey, there's no thymine on RNA. Let's say uracil side there. Okay, I think that makes sense. All right, so complementary here would be that, I believe. I did that right. Okay, so and then what would the strand, the opposite strand of the DNA be? It would be GGATC. So that's why we call this the coding strand, because the messenger RNA here, you can see it looks the same as the coding strand. So anyway, the terminology, again, can be a little bit confusing there. So the RNA polymerase is moving down that template strand. It's adding triphosphate ribonucleotides. So we saw for DNA polymeration, remember it was triphosphate deoxyribonucleotides, carrying with them their own energy for the polymerization reaction. Okay, let's think a little bit more closely about this enzyme RNA polymerase before we think about how it is that it initiates this transcription. And I think it's good to compare it with the enzyme of replication. So it was DNA polymerase 3. That was the enzyme, the main enzyme that was used in replication, and RNA polymerase for transcription. All right, so we talked about in replication that DNA polymerase 3 needed another enzyme to break the hydrogen bonds of the complementary base pairs, and we call that helicase. The RNA polymerase actually does that on its own. Okay, unzips. It unzips the DNA itself. Okay, so it doesn't need another enzyme to go ahead of it and pry open that DNA. We saw that DNA polymerase 3 needed a primer because it couldn't start all by itself. Not the case for RNA polymerase. We don't need a primer here. Okay. Of course, DNA has the thymine base, RNA has uracil. And we mentioned that DNA polymerase 3 has really good proofreading function and that it moves really fast. I don't think I wrote in the number how often it makes a mistake, but the error rate is about 1 in 10 billion. So that's good. We need, if we're going to copy the blueprint or copy the recipe, we need that to be really high fidelity because, you know, every time you make the recipe, you're going to make a mistake if that recipe is wrong. The RNA polymerase proofreading is not as good. It does make more errors, about 1 in 10,000 base pairs, it will make an error. But again, that's probably okay. The next time it's transcribed, that DNA strip, it will probably be correct. So it's not as much of a problem if a few more errors are made. Okay. All right, so another figure that I got from another source, but I don't think your book even, it doesn't, I don't like the figure that it has for initiation. It's not correct, it just doesn't have much detail to it. So this, it's oftentimes we just draw a line for DNA. All right, so just keep in mind, this is the DNA, the one strand that's being used as a template for the messenger RNA. And this sequence of bases kind of, we call this upstream of where transcription is going to start here, you know, about right here. Sometimes we call that the initiation site. Upstream, meaning in this direction to the left of that initiation site, we call this sequence the promoter. Okay, so this is a sequence of bases that we know that's where the RNA polymerase has to bind. All right, so here's our RNA polymerase. And one principle that I want to try to get across to you is that the more tightly RNA polymerase binds, the more likely the DNA or the genes will get, gene or genes will get transcribed. Okay, so there is a particular protein in prokaryotes that is necessary to come and bind with that RNA polymerase. RNA polymerase is a great big huge enzyme. When it's coming to bind to the promoter region, this protein called sigma factor, I just learned not that long ago that the Greek alphabet has capital and lowercase letters. So this is a lowercase sigma. Okay, sigma factor. Again, this is just a protein, helps RNA polymerase bind to the promoter. And there's actually, a cell will have different sigma factors. Some sigma factors help the RNA polymerase bind really well. Some sigma factors make it bind less as well. So it might not be in the cell's best interest to always have a gene transcribed. It might be a protein that's not really, you know, instructions for a protein that's not needed that much. So it may make sense for that only to get transcribed every once in a while. But that's what that sigma factor does. It attaches with the RNA polymerase just during initiation, and then it falls off. Two other regions that are important, -10 base pair region, that region, as well as the -35 base pair region. We think those 5, 6 base pairs are sites that are especially important for recognition. So the RNA polymerase especially recognizes the -10 and the -35 regions. So that -10 region is so important that we have a special name for that. We call that the Pribnow box. In eukaryotes, we call it the Tata box, which you may have talked about that in another class. It kind of makes sense because it's often a region that's rich in thymines and adenines on the DNA. Minus 35 region, to my knowledge, doesn't have a special name. We just call it the minus 35 region. Okay, so again, those regions are especially important for the RNA polymerase to recognize. All right, so I want to keep looking upstream here for just a moment, and this slide looks more complicated than what we really need it to. But to me, this sort of helped me make some connections when I kind of understood this. So what this shows, all these, remember DNA, it's a sequence of bases, right? And this is the sequence of the promoter regions of several different operons in E. coli. So remember what an operon is. It's a group of genes under control of a single promoter. Okay, a group of genes under control of a single promoter. So we talked about how bacteria can be polycistronic. So there can be a number of genes that all get transcribed at once. Okay, eukaryotes, monocistronic, usually just one messenger RNA gets made into one protein. Okay, so here's some different operons. Lac operon, by far the most famous one. We're going to talk about that one a little later, as well as the trip operon. I just looked it up before class. There's actually, we think, between 600 and 700 different operons in E. coli. So yeah, that's a lot. I don't know that they've all got figured out exactly what they do, but looking at the sequence, that's what they think. Okay, and this, I like this figure because, again, here we see the -10 region and the -35 region of that promoter is highlighted. And down here, you'll see the consensus sequence. So basically, they've looked at those -35 regions and -10 regions of a bunch of different operons, and they've noted that most often, the first base pair is a T, the second is a T, the third is a G. Okay, that seems to be the most common base that's there. And they think that the more closely this 35 region, -35 region is to this consensus sequence, the more tightly the RNA polymerase will bind. Okay, same thing with the -10 region. The closer it is to these base pairs, then the more tightly the RNA polymerase binds. And again, the more tightly it binds, then the more likely that gene is to get transcribed, made into messenger RNA. Okay, so that's just what I wanted to point out with that. Okay. So let's say the RNA polymerase binds tightly and it starts to transcribe the gene. So elongation is fairly straightforward. This is your picture from your book. Here we see the sigma factor dropping off. It's not necessary anymore. The RNA polymerase is, again, breaking apart those hydrogen bonding between the two strands of DNA. It's only transcribing one. It's adding on the triphosphate ribonucleotides. And it stays kind of bonded temporarily to the DNA strand. Here you can see the 5' end is already fallen off the DNA template. And the directionality. So just like in replication, this is the 3' end here. There's a 3' hydroxyl group there that the new ribonucleotides are going to attach to. Okay, so the new strand is always growing in the 3' direction. That's complicated because sometimes a question might say, it's moving in a 3' to 5' direction, but you're talking about the template strand. So if you can just keep in mind that the new strand is always adding on to the 3' end, then that will serve you well. All right. So like I said, there's a particular stopping point we'll get to termination in a minute, but I just wanted to point out that if this is a stretch of DNA that's really important, that the cell needs a lot of whatever protein that's encoding for, then you can have lots of RNA polymerase is just as soon as one binds the promoter and starts the transcript, another one comes in. Okay, so it can get transcribed really quickly. Okay, and both in eukaryotes and prokaryotes that can be the case. All right, there's two different ways that we know of in bacteria that transcription gets stopped. And we think that they both happen with about equal percentage. So maybe 50% one, 50% the other. The first method is dependent on a particular protein called Rho. So we call it Rho dependent. Okay, and this is actually a helicase enzyme. Remember what helicases do? They unzip the DNA, break those hydrogen bonds. So the basics of this is that the Rho protein just moves down along the strand of this new transcript and breaks these kind of temporary hydrogen bondings that take place with that DNA strand, and eventually just dissociates the messenger RNA from the template strand, and transcription stops. Okay, so here we've got the messenger RNA is falling off, and to me, this is not terribly satisfying. Like, how does the protein know when to do that, and how does it do it at the right speed? Your book just presents it in a pretty simplistic way like this. The more that I read about it, it gets extremely complicated, and we don't really understand everything about it. So one method involves a Rho protein, and again, it just dissociates that transcript. Okay, that's the basics of that. The other method does not involve the Rho protein, so we call that Rho independent. It's also known as sequence dependent or self-termination. So this method of termination of transcription goes by 3 different names. All right, and kind of just got to recognize all of those. Okay, the sequence dependent to me kind of makes more sense. That means that it's dependent on the DNA sequence. So remember, here's the DNA down here at the bottom. The new RNA transcript here is in red. And basically, there are sequences in the DNA that are inverted repeats. Okay, they also can be called palindromes. And what that means is when the RNA is transcribed here, basically this sequence on one part will match up complementary to another part. And so the RNA, when it dissociates from the DNA, kind of curls around into a hairpin loop. Oh, there, it's written right here. Okay, and the mechanical strain imposed by that hairpin loop causes the new transcript to fall off of the DNA. Okay, so again, there's some, we'll call it mechanical strain imposed by that hairpin loop. In addition, you'll see here, there's a sequence of lots of adenines on the DNA, which corresponds to lots of uracils on the RNA. And remember that uracil adenine, there's only 2 hydrogen bonds between those. The G's and C's, there's 3 hydrogen bonds. So this probably would fall off more easily. Okay. So again, hairpin loop caused by, termination by the sequence in the DNA. And I did post this other figure from another textbook. Oops, I didn't cite that very well. This just explains a little bit better what an inverted repeat is. So if you're trying to kind of understand that in your mind, Sometimes we call those palindromes, but a palindrome in DNA is not quite like a palindrome in English. Like, you know, the name Hannah is a palindrome, the same forwards and back. An inverted repeats is like here and here, they're going to be complementary to each other when they match up. So if you're interested in looking at that a little bit more closely, I just thought that made it a little bit easier to understand. All right, so that is transcription in a nutshell. Okay. I always think it's good to, you know, like I explained maybe last lecture, we're looking at the genetics here from a prokaryotic perspective, but I want to pull in maybe what you've learned in other classes, biochem or genetics, about how, you know, the same process works in eukaryotes. So it's always good to kind of compare what we learned with maybe what you know, or just to kind of help give you some perspective. So where is this process taking place? Transcription. Well, the answer is it's taking place where the DNA is. Okay, so in prokaryotes, the DNA is going to be in the cytoplasm. And we've talked about how that's in a kind of a special nucleoid region, but it's not bound by any kind of membrane. So we still consider that to be in the cytoplasm. And of course, eukaryotes have their DNA in the nucleus. Okay, so kind of keeping track of where this is taking place is always important. The complexity is a little bit different between eukaryotes and prokaryotes. It's a lot more complicated in eukaryotes. Eukaryotes have lots of transcription factors, special proteins that have to be in just the right place. They have lots of elongation factors. In eukaryote transcription, they have a special RNA polymerase for each kind of RNA. So they have an RNA polymerase for messenger RNA, an RNA polymerase for ribosomal RNA, one for transfer RNA, where prokaryotes just have one enzyme that makes all different kinds of RNA. All right, so there is a difference in complexity. And I do want to mention there is some processing in that transcript that has to take place for eukaryotes. And that's, again, after termination of transcription, the addition of a 5' cap. So it's a 5' modified guanine cap. And again, this is in eukaryotes only. That gets added after termination, as well as polyadenylation at the 3' end. Okay, so lots of adenines are added on to the other end. And I mention that because it makes sense. Those two things are done to stabilize the RNA. We're particularly interested in messenger RNA, so we'll say messenger RNA. Because the messenger RNA is going to need to travel for eukaryotes, because it needs to travel from where the DNA, where it was made, to the ribosomes. Okay, so for eukaryotes, it has to travel from the nucleus out to the ribosomes. And there's lots of enzymes in the cytoplasm that would degrade the ends, right? So the 5' guanine cap and the polyadenylation helps protect the ends of that messenger RNA transcript as it travels. In prokaryotes, pretty much everything happens right in the same spot, so that's not as necessary. And lastly, in eukaryotes, there are some of the DNA is transcribed that really doesn't do anything that we're aware of. It doesn't actually get made into a protein. Some of that messenger RNA gets spliced out. Okay, so the introns, that stands for intervening, I don't know if I spelled that right, sequences. That gets cut. And then the exons stands for expressed genes, or that's the way I remember which is which anyway. That stays, remains, we'll say. Okay. So there's actually sort of a pre-messenger RNA transcript and then the introns get spliced out of that. So I really like this figure that's in your book, 711. Again, this is processing in eukaryotic messenger RNA. Here you can see the 5' cap on that transcript, the poly A tail. There's a little further processing by some kind of protein complex that we call a spliceosome complex that comes in and gets rid of the introns. And then that messenger RNA is ready to leave through a nuclear pore out into the cytoplasm where the ribosomes are. Okay. And I just pulled this from another source. I just wanted to point out that splicing is not done the same way every time. We can actually get different proteins from the same gene depending on how it's spliced. All right, so here, if the introns are the line and the exons are the blocks, you would expect kind of all the introns to get cut out, and you would be left with this block pattern on the left. But if, say, maybe you had a big cut right here, you might lose one of those blocks and have a unique protein there. Or over here, the one on the left, maybe part of this block was spliced out, so you have a little different here. It would give you 3 different proteins. Sometimes that happens purposely. Sometimes there are errors in splicing that give us misformed proteins. And we know that there are diseases that are errors in splicing. Duchenne's muscular dystrophy is one. I believe Parkinson's is another. So again, but that's unique to eukaryotes. Okay. All right, so what happens next to this transcript? It's going to get translated into a polypeptide at the ribosomes. So the steps of translation kind of look like the steps of transcription. It's easy to get transcription and translation mixed up. So you want to try to keep those processes separate in your mind. The tRNA, remember the transfer RNA is what brings the correct amino acids to match up with the messenger RNA transcript at the ribosomes. When the tRNA is carrying, when the amino acid is attached to it, we say that it's charged. Okay, so tRNA attached to its appropriate amino acid. Okay, so that's I'm not going to go through that process. That's mediated by a couple of enzymes, requires a little ATP. Your book skips it, so I'm going to skip that as well. Okay, but just know that that's, when the tRNA by itself is uncharged, when it's carrying its amino acid, then it's charged. Okay, and then kind of like transcription, we go through an initiation, elongation, and termination. And then I'm just going to mention a couple of things that may have to happen to that protein before it can be functional, just to make you aware. All right, so to understand translation requires some appreciation or some understanding of the genetic code. And this was elucidated by a lot of trial and error. It required radioactive labeling, sequencing, cumbersome processes. back in the early 60s. 1961 was when Marshall Nirenberg elucidated that. He was working at the NIH at the time. And so they were trying to understand how the messenger RNA carried the instructions for proteins. And at first they thought, well, okay, it's not one base to one amino acid because there's only 4 bases and there's 21 amino acids. If it was a combination of 2 different bases, that wouldn't make sense either because there's only 16 different possible combinations if you have 2 bases. So then they reasoned, well, it must be 3. It must be a set of 3. Of course, there's 64 different combinations of 4 different bases, 3 of them together. So there is some redundancy in this. In other words, sometimes it doesn't matter what that 3rd base pair is. So for example, like for leucine, it doesn't matter at all what that 3rd base. So we say there's some wobble in that 3rd base pair. We also describe that as being degenerate. And usually that's not a very nice thing to say about somebody if they're a degenerate, but that just means that it doesn't really have a particular order. If it's degenerate, it's lacking order. So all 3 of those are ways to say that it doesn't always matter what that 3rd base is in the codon. Okay, so every 3 bases in the messenger RNA is referred to as a codon. So again, this is messenger RNA. Okay, and again, there's over 20 amino acids here. I wouldn't expect you to have this memorized for sure. Probably should recognize the start codon. So AUG, that's always the first one. The first amino acid to begin a protein is always methionine. Okay, so that's where it starts. And there's 3 different stop codons. So when those codons are read at the ribosome, that causes the termination of the newly formed protein. Okay. So how does that start in prokaryotes? So remember, your book uses this, little blue bent thing to signify the transfer RNA. Okay, and the messenger RNA here is going to line up at the ribosome, starting at the 5' end, and the next graphic will make it a little bit more clear, I hope, why that has to be the 5' end. But it has to line up just right at the small ribosomal subunit. And that first, so here's our start codon, AUG. That codon encodes for a tRNA with the anticodon that will be complementary, and that's carrying, it's actually in bacteria formal methionine. So it's in eukaryotes, it carries methionine. It's got a little formal group on it for bacteria. And that's going to come in at the site on the small subunit that we call the P site, or the peptidyl or peptide site. Okay, that's where the first one comes in. Okay, and that has to line up just right. There's some energy that has to be used up in that reaction in the form of GTP, very similar to ATP. Okay, once that first amino acid comes in and binds at the P site, then the large ribosomal subunit comes and binds and makes that initiation complex. Everything comes together once the first one is there. Okay. So looking at that process in a little different way, which at least for me, this was helpful. I got this from a different textbook. Here's the strand of messenger RNA. Okay, and I said that it has to line up just right with that small subunit of the ribosome. And in this picture, for some reason, they drew that small subunit on the top instead of the bottom. That's kind of the way you normally see it. And remember what makes up ribosomes. There's proteins there, and then there's strands of ribosomal RNA. So here it's drawing in some ribosomal RNA, and there is a sequence on the messenger RNA that has to line up just right with that 16S ribosomal RNA. So that sequence is really important. When a sequence is important, we just like to give another name to it. And here we call it the Shine-Dalgarno sequence or Shine-Dalgarno element. And they do mention that briefly in your book. More often, they will call this the ribosome binding site. Okay, so that's a sequence on the messenger RNA that has to bind with the ribosome. And then the ribosome has a messenger RNA binding site that has to bind with the messenger RNA. Okay, and it's really important that that lines up just right, because it has to line up just right so that that AUG falls into that P site. If it's off by one or the other, then it's going to read it in the wrong frame. So then you're going to have UGC. Well, it doesn't code for anything. It's not a start codon for sure. So initiation might not even start unless this is lined up just right. Okay. And then how I remember that it's lining up with the 5' end. Well, remember in prokaryotes, we can have transcription and translation going on at the same time. So here's my little drawing up here, adding to this figure. It's a little bit primitive, but here's the DNA and RNA polymerase that's transcribing. Well, you know, it's always the RNA is always adding on to the 3' end. If the end of that transcript is funneling right into a ribosome, that's going to have to be the 5' end. Okay, it can't be on at both ends at once. Okay, so let's see. I think I wanted to. Okay, so a few notes there. Transcription and translation can happen simultaneously. Okay. And that Shine-Dalgarno sequence lines up, let's see, on messenger RNA, lines up with the ribosomal RNA, or the small subunit of the ribosome. Okay, so sometimes we get confused. Remember the Pribnow box, that's the -10 region. That's important for initiation of transcription. Shine-Dalgarno sequence, important for initiation of translation. Okay. And then we'll go back to your book's figures that talk about peptide elongation. So the initiation complex, the small subunit and the large subunit of the ribosome have come together. Here we have a new transfer RNA, new charge transfer RNA coming into the A site. You can call that the amino acyl sites. I like to just call it the acceptor site. Okay, all new tRNAs come into that A site, except for the first one. The first one starts at the P site. All the rest of them come into the A site. The peptide is just briefly transferred over. So you can see here that it's going to sit really briefly at that A site. The peptide bond is formed by ribosomes. Remember last time we said that ribosomal RNA can act as an enzyme. So it makes those bonds between the amino acids there briefly in the A site, and then it switches over to the P site. So we say that the P site holds the growing peptide. Okay, the beginning chain of amino acids kind of really gets held at the P site. Now this very first tRNA gets switched to the E site or exit site, and it's dissociates and goes off and gets charged again with another formylmethionine. Okay, so a continuation, like we could keep going with this. Here's another tRNA carrying another amino acid, threonine comes into the A site. The peptide is transferred briefly to that A site, and then everything kind of shifts over to the next site. So we've got this growing polypeptide again at that P site. And this all happens incredibly quickly. About 15 amino acids are added per second. It's kind of mind-boggling how quickly that can happen. How do they get matched up? How's just the right one get matched up? This is a nice animation that I found on YouTube. It kind of helps you appreciate how complicated it is. Not in real time still, but it shows it happening pretty quickly. So take a look at that if you have a chance. Start at about the 2-minute mark. I think it was showing something different before that, and it's just a couple minutes long. Okay. So you can also just keep in your mind, you can have one transcript. Here's messenger RNA transcript with lots of ribosomes on it. So as soon as one ribosome clamps on, as soon as that's fed through there, another one can clamp on. So we call that a polyribosomal complex. That can happen in both prokaryotes and eukaryotes. I think it's more likely to happen in prokaryotes. Okay. And how does the process stop? Well, a stop codon is read on the messenger RNA at the A site. So your book doesn't, I don't think, it may now have a graphic for this, but I found this somewhere else because originally the book didn't. So here's UAA, that is one of the stop codons. There's no tRNAs to recognize that stop codon. So there's kind of a pause, release factors. So again, there's kind of a pause there. Release factors, some proteins called release factors will come in and bind to the A site instead. Okay. And then that's going to activate ribozymes to cleave off the peptide and release it from the A site. Sorry, P site. Okay. And then the whole ribosome is going to dissociate. Okay, split into its small subunit and large subunit. Okay, so in transcription, termination either required a row protein or it was dependent on the sequence. Translation requires a stop codon and some release factors. Okay, so that's the gist of that. So after you've got this new, a cell has made this new protein, okay, typically that protein is not going to be functional right away. It could be, but almost always that formylmethionine is cleaved even before translation is done. I'm not really sure the significance of that, why that gets removed. If it's an enzyme, there may need to be some kind of cofactor that's added or coenzyme before the enzyme is functional. Oftentimes there's some folding that has to happen with some hydrogen bonding perhaps, or maybe a couple different proteins have to come together, be bonded together before it makes a functional protein. And especially for prokaryotes, it may not be beneficial for the organism to have that protein functional right away because it might be something that's designed to harm something else. It could be a toxin. Okay, so they don't want it functional right away, there may need to be some kind of cut that happens or trimming that might need to happen right before it's secreted outside of the cell. Of course, eukaryotes can make proteins and then, you know, enclose them in a membrane and then get them out of the cell that way. Bacteria can't do that, right? They have that thick cell wall that they can't endo and exocytose things. All right, so they don't want the toxin to be active inside the cell. They want to release it and have it act on something else. All right, so I kind of like this figure. For some reason, they left this off in the newest edition of the book, or they simplified it. But to me, I like the way this kind of helps you put things together. How you've got the template DNA strand, and maybe what the messenger RNA would look like. You know, writing in the start codons and the stop codons and all that, and how you can get perhaps several different polypeptides from, you know, different genes that all get transcribed together. Okay, so to me, you know, I like that figure. Kind of helps me think about what's really going on there. And once again, I want to compare translation in eukaryotes to prokaryotes. Okay, just kind of reiterating some of those things that we just talked about. Remember, prokaryotes, the messenger RNA can be, it's not always, polycistronic. Okay, so like I just said, okay, we can get several different proteins from one transcript. In eukaryotes, we talked about the 5' guanine cap and the 3' polyadenylation. In eukaryotes, it's that 5' guanine cap that lines up just right at the ribosomes. For prokaryotes, they have that Shine-Dalgarno sequence. I mentioned this already too. Transcription is going to happen in the nucleus where the DNA is for eukaryotes, translations at the ribosomes. So transcription and translation in the cytoplasm at the ribosomes in the cytoplasm for translation. And just again, I don't really know the significance of that. If there is one, the first amino acid is methionine for eukaryotes and formal methionine for prokaryotes. Actually, archaea have methionine, so I didn't put that absolutely accurate, but so it's kind of interesting. The archaea are a little bit more like the eukaryotes in some ways. Okay. All right, and you know, like I said, for metabolism, I showed you a table that was in your book. Sometimes these tables are nice to just kind of cover up and quiz yourself. I will say we did not talk about the directionality here of the amino acids, so you don't need to worry about that. But anything else, you know, it's very common, again, to get mixed up between the processes, what's taking place in replication, what's taking place in translation. So it's kind of a nice tool just to kind of cover up and quiz yourself. Those basics are important. Okay. So with our time left, let's talk a little bit about regulation. Okay. So this refers to how does a cell control how much of proteins actually get made. It's really important for a cell to regulate that because it's quite expensive energetically to make a protein. Okay, for the average protein, which is about, say, 300 amino acids long, how many ATP do you think it costs the cell just to translate that protein? Any guesses? Okay. It's about 1,200 ATP it costs the cell just to make one average length protein. Okay, so the cell is going to want to only make proteins that it absolutely needs. So it's going to regulate in order to conserve energy. Okay, it just doesn't want to spend that energy that it doesn't need to. Okay, and in E. coli, about 90% of the cell's energy is devoted to making proteins. All right, so there's different kinds of regulation. We talked about this, that kind of complicated slide where the sequence of the promoter regions of the operons. Depending on that -10 and -35 region, the sequence there will help depend how tightly the messenger RNA binds to that promoter region. And again, if it doesn't bind very tightly, then it's the proteins, the transcripts, not as likely to get transcribed. So a cell can actually vary a little bit that sequence, and that might depend how often the gene gets transcribed. I mentioned before that there's different sigma factors that a cell can use that will determine how likely that transcription is to take place. But we're going to talk a little bit more here about operons. And again, this just means that genes are regulated together. And this is mostly a prokaryotic thing. Eukaryotes have some gene clustering that almost resembles an operon, but we think that that's very rare in eukaryotes. So again, this is kind of a prokaryotic thing. It's a very efficient way to get just the right number of protein in. And typically, they're proteins that maybe all are necessary in a particular pathway. So it makes sense to kind of get the same amount of all of them. You wouldn't want to have more of one and less of another because they kind of are all working together. So if we look at the parts of an operon and review that a little bit, of 3 different operons that I want to look at that have that are regulated a little bit differently. Remember the promoter region. This is where the RNA polymerase binds. Okay, what we're regulating whether transcription takes place here or not. So that's where the RNA polymerase binds. The operator region then is where a repressor binds. Okay, and that's easier to remember if you understand that the repressor would be a physical block that would keep the RNA polymerase from moving down the transcript. Okay, if there's something here to stop it, it's going to repress the transcription. Okay, there's a few structural genes, 3, 4, 5, whatever, there's difference there. And usually there might be some regulatory gene upstream here that would actually transcribe any repressor. Okay, so that's kind of the basics of an operon. Okay. Your book doesn't quite, the book actually cut down what it talks about in operons in the last edition. And it doesn't organize it quite in the same way. It talks about positive control, but doesn't really mention the term negative control. So it makes sense, though, to me that if you've got positive control, you've got negative control as well. If an operon is under negative control, that just means that there are specific repressor proteins that are important in controlling that. Okay, and like I said, the repressor is always going to bind to the operator. There's 2 different kinds of negative control, and your book talks about this as well. There are inducible operons as well as repressible operons. And it's kind of, to me, I have trouble keeping those straight, but an inducible operon is usually off, meaning it's not transcribing those proteins. Okay, and it's only going to transcribe them if there's some inducer present. So the inducer would deactivate the repressor. And these are usually catabolic pathways that are controlled this way. So pathways that involve the production of proteins or enzymes to break down some metabolite like lactose, okay? So the lac operon is an example here of an inducible operon. We'll look at that in just a minute. An example of a repressible operon is that trip operon. This is regulating the enzymes that are necessary to biosynthesize tryptophan. So it's an amino acid that bacteria need. Most of them can make it themselves. Okay, so usually anabolic pathways, biosynthetic pathways are regulated this way, and they are usually on. So they are most often going to be transcribing these proteins because, because they need to synthesize the amino acid tryptophan. Okay, and they're going to involve a co-repressor. All right, so let's look at an example of both of those before we turn to positive control. The lac operon should be fairly familiar. I know it's one of those things that you learn for the exam and then you try to forget again as quick as you can. See if you can, I came across this diagram, online. See if you can label what each one of these things are. Okay, especially after we just kind of reviewed what's what. See if you can label it. So I'm going to give you a minute to do that. A little breather for me talking here. Give you a minute and see if you know, well, what's number one, what's number two, see if you can reason it out. I'll write down the parts here and you can just kind of match them up. All right, let's see how you're doing. So let's see, in the order that number one, that's the RNA polymerase. Number two is a repressor. The RNA polymerase is going to be bound to the promoter region, and the repressor is going to be on the operator region. These little snowflakes here, that represents lactose or allolactose. Pretty close to the same thing, just a little different linkage between the two monosaccharides there. And then I'm not really worried that you know the order of the proteins. Lac A, that's 8, that encodes transacetylate, thiogalactoside transacetylate. We're not really quite sure what role that plays. It might be some kind of detox role. We're not really sure. Lac Z, let's see, that's the first one. That's beta galactosidase. That's the enzyme that splits the two monosaccharides that make up lactose, glucose, and galactose. And 7, lac Y, encodes for lac permease. That is what enables the lactose to come into the cell very quickly. So it's kind of like a fast pass, or if you've ever traveled in Chicago, you've got a fast pass, you can just go right through those toll gates. You don't even have to slow down. If you don't have a fast pass, you got to stop and pay the toll and get change and all that. So lac permease is like a fast pass for lactose. You can get into the cell really easily. It can still get in, but it's just a little bit more cumbersome. All right, so these genes, especially the beta-galactoside and lactofermiase, and maybe the other protein there to some extent, are necessary for a cell to use lactose as an energy source. Okay, so it only wants to turn those things on if lactose is present. Okay, so if lactose is present, it acts as an inducer. It binds to the repressor, takes it off the operator, and now the RNA polymerase can transcribe the gene. That physical block has been pulled away. Okay. And then there is another layer of control there that we'll come back to. So it's not quite this easy. Remember, a cell is always going to choose to use glucose first. So even if lactose is there, if glucose is there, it doesn't want to use the lactose. So we'll get back to that layer. Probably we'll do that next time. The trip operon is an example of negative control. Remember, this is a repressible operon. A really nice animation I found that explains this really nicely, has a couple quiz questions. This is one that I was having trouble getting this to play because my Flash player was out of date. So if you have trouble finding this particular one, there's probably some other good ones. But with the lac operon, so there's actually 5 different genes that encode for 5 different enzymes that are necessary for tryptophan biosynthesis. Okay, so they're all regulated together. That makes sense. And if a bacteria is in medium without tryptophan, then it's pretty much going to need to be making this all the time. Okay, but if we're growing this organism in a medium where we've added tryptophan to it, it's not going to make sense for the organism to keep making it because it's already there and readily available. So it wants to conserve energy. That tryptophan actually acts as a co-repressor, binds to an inactive repressor, and now when the repressor is active, it binds to the operator and stops the RNA polymerase, keeps it from transcribing the gene. Okay. Right. I think maybe this is a good place to stop because we're a little ahead of ourselves anyway. I want you fresh thinking about positive control. So we will finish up chapter 7 next time and start viruses on Monday. Woohoo! All right, so have a good weekend.